Designing experiments for stem cell research requires careful planning to ensure reliable results and ethical standards. Here's a structured guide on how to approach the design of experiments in stem cell research. Understanding stem cell research. Stem cells have the unique ability to differentiate into various specialized cell types, making them valuable for studying development, disease modeling, and regenerative medicine. Experimental design in stem cell research aims to manipulate and study these properties effectively. Steps to design experiments in stem cell research. Research objective and hypothesis. Clearly define the research objective and formulate a hypothesis that addresses a specific aspect of stem cell biology or application. Determine whether the study will focus on pluripotent stem cells, e.g., embryonic stem cells, or multipotent stem cells, e.g., adult stem cells. Experimental design. Cell culture conditions. Establish optimal culture conditions to maintain stem cell pluripotency or differentiation potential. Consider factors such as growth factors, media composition, and substrate properties. Experimental groups. Define experimental groups based on the variables being tested, e.g., differentiating agents, genetic manipulations. Control groups. Include appropriate control groups to compare experimental outcomes against baseline conditions. Controls may include untreated cells, vehicle-treated cells, or cells under standard culture conditions. Assessment methods. Cellular characterization. Use techniques such as flow cytometry, immunocytochemistry, or gene expression analysis to characterize stem cell populations and monitor differentiation. Functional assays. Conduct functional assays to evaluate stem cell functionality, such as colony-forming assays, cell migration assays, or in vitro differentiation assays. Molecular analysis. Perform molecular analyses, e.g., PCR, RNA sequencing, to examine gene expression patterns and signaling pathways involved in stem cell fate decisions. Ethical considerations. Compliance. Ensure compliance with ethical guidelines and regulations governing stem cell research, including informed consent for human-derived stem cells and adherence to animal welfare regulations for animal models. Use of human stem cells. If using human stem cells, ensure adherence to guidelines set forth by ethics committees or institutional review boards, (ERBs), regarding consent, privacy, and the ethical use of embryonic stem cells. Data analysis and interpretation. Statistical analysis. Apply appropriate statistical tests to analyze experimental data and determine statistical significance. Data interpretation. Interpret results in the context of the hypothesis and existing literature. Discuss implications for stem cell biology, potential applications, and future research directions. Considerations for advanced techniques. CRISPR, Cas9 genome editing. Utilize genome editing techniques to introduce precise genetic modifications in stem cells for studying gene function or correcting disease-associated mutations. Organoid and tissue engineering. Develop three-dimensional, 3D, culture systems or organoids derived from stem cells to model tissue development, disease pathology, or drug screening. Future directions in stem cell research. Single-cell analysis. Incorporate single-cell sequencing technologies to investigate cellular heterogeneity within stem cell populations and during differentiation processes. Bioinformatics and computational modeling. Integrate bioinformatics tools and computational modeling to predict stem cell behavior and optimize experimental outcomes. Conclusion. Designing experiments for stem cell research requires a systematic approach to address research questions, manipulate stem cell behavior, and interpret complex biological data. By applying rigorous experimental design principles, adhering to ethical guidelines, and embracing technological advancements, researchers can advance our understanding of stem cell biology and harness their therapeutic potential in regenerative medicine and disease treatment. As stem cell research continues to evolve, innovative experimental approaches will drive discoveries that shape the future of healthcare and biomedical science.